We basically found that there are a number of proteins, and in particular a protein called ID2, which is a protein that is normally operating in normal stem cells, that in fact is activated through a very convoluted network of molecules in hypoxic cells, in hypoxic tumor cells, to drive this cancer stem cell state. Now the interest, the particular translational interest of this network is that we discovered that the ID2 protein completely disrupts what is known as the most important tumor suppressor pathway, a pathway that suppresses normally tumor growth and is there to limit the effect of hypoxia. This pathway is centered around a gene called VHL, the von Hippel-Lindau, and we showed that when ID2 is activated by hypoxia in these cancer stem cells, it completely disrupts this VHL ubiquity ligase pathway. The function of VHL is to degrade, to eliminate other proteins, to destroy other proteins, and in particular a family of proteins called HIF that are really at the core of aggressiveness of these cancer stem cells. So we essentially dissected the entire network, and because this has been done in a very sophisticated molecular analysis, now we know exactly how we can design drugs that will prevent, for example, the interaction between ID2, this bad protein, and the VHL, the ubiquity ligase tumor suppressor complex. So what I presented today is the proof of principle that some drugs that have been generated from this molecular screen that we have done based on our findings can actually prevent this disruption of the VHL complex, therefore indicating that this might be extremely specific and promising compounds to basically target exactly specifically the most aggressive cancer stem cell population that is generated by this ID2 VHL network.